and a warm welcome to our webinar today, which is about a step-by-step guide on how to develop your own organ model. So we will start with a bit of housekeeping. On your left-hand panel, you'll find a Q&A section where you can submit your questions throughout the webinar or after. And you also have a chat function if you've got any general questions, like require any files or stuff. I can always send this. Also, to let you know at the beginning, we will send the recording after the webinar. So yeah, if you want to share it with your colleagues or revisit it, that's absolutely fine. So before introducing our speakers, I would like to share a poll because we would like to understand a bit where you found out about this webinar. So you can vote on where you found this. So while you're completing this, I would like to introduce our first speaker for today, which is Joanna Novaka. She developed a microtic heart-on-chip model during her PhD using iPSC-derived cardiomyocyte subtypes to study signal propagation and evaluate the efficacy of cellular transplantation. Her project was carried out by a multi-biotech collaboration with the University of Tübingen and combined advanced in vitro modeling with intensive academic industry networking. Cool. So now that we learned a bit about Joanna, we would like to know a bit about you. So my next call is about which application or organ model is of interest for you. Okay, so while you're doing this, without further ado, I'm handing over to Joanka.
Thank you, Juliana. It was a very nice introduction. Yes, so my name is Joanna. I'm here as a lead scientist at Dynamic 42 for the development of the stem cell based models on a chip. And today I would like to present to you how to start with organ on a chip. So once you're actually thinking that organ on a chip would be something interesting and something that would be fitting for your study, but it still feels a little bit overwhelming. To start with it, today I would like to present a little bit guides or step-by-step how to build up your model. But first I will start with explaining who we are. We are a small company established in 2018 as a spin-off of University Klinikum Jena in central Germany, East Germany, in Thuringia. We are developing organ on a chip. We are producing and we are selling the organ on a chip technology, but we are also acting as a fee-for-service company that runs the internal research for you. Okay, so the next hour will be all about organ on a chip. But what exactly is an organ on a chip? If you would ask an AI, which is a very fashionable thing to do in 2025, we all ask AI different stuff, but if you ask AI what is organ on a chip, it does not really get it. So as you can see, well, the definition of organ on a chip is not a musical instrument on a pack of food, but rather if you ask a scientist, is this biochip connected to tubing, reservoirs, microfluidic pumps, peristaltic pumps, cells, something all about this. So since AI doesn't get it, so I understand if you feel that this is a little bit overwhelming at the beginning. But don't worry, we are here to answer some questions. For example, how our chip is actually, how our system is actually built up. Here you can see a biochip. We here at the bottom, laying on the bottom of the incubator. The biochip is tagged to our tubings and reservoirs that allows to perfuse the media with the usage of our peristaltic pump. So this is an example of image of if you would be ending up with using a biochip, this is how your incubator and daily work would look like. What is actually inside of the biochip? There are two channels. There's every channel has an inlet and outlet. There's also a cell culture chamber, which is transparent and allows you to use the microscopy study to see your culture. Our chip is built of two separate chambers. So one chip is composed of two independent microfluidic systems. So you can run two technical duplicates on one chip. Inside, when you look through this transparent part, we call it chamber on a chip, you can see two channels, one top channel and then the channel laying on the bottom channel. Between those two channels, there is a porous membrane. Yes, you can see the pores. You can see, so with the top chamber, you can cultivate one type of the cells, and on the bottom chamber, you can cultivate the other type of the cells that will be spatially separated, but still the nutritions from the upper chamber can flow through to the bottom chamber and affect your cells. So with Dynamic 42, we established already some organs. So we have applications for liver, intestine, lung, cancer, and vasculature, and more are actually coming. So if you would be interested in the future applications, you can go ahead and follow us on LinkedIn. Okay, so we know a little bit about the chips, but still knowing a little bit about the chips, having them on our computer screens feels a little bit overwhelming when you think that you actually have to bring this technology for the first time in your lab. But from my experience, my personal experience, from the experience of our customers, this is really just the feeling that you have only at the beginning. And I hope that now I can share a little bit of my experience with the chips, and I can share with you how to think about all this process. And maybe it will be easier for you to establish or organize your future experiments. So let's go through it step by step. Let's start very easy with the organ. So the way that I will be going on with this presentation is I will be giving you this step-by-step guidance with a very general examples, but I would like you to be a little bit more interactive with me. And I want you to think about your own study, your personal study, and how these steps can be applied to your study. And if you can go through all the steps and actually visualize or have a feeling that this is something possible for you in your research, and if actually everything is fitting together into this final picture, or actually if you have some problems with some steps, then please go ahead and ask some questions, and I am here to guide you through those also more difficult steps. But let's just start easy. So I think everybody will have the organ of choice that you would like to study. So, and obviously a lung is not the heart, it's not the brain. So with organ on the chip, we have to work with one organ on a chip, on a single chip. I can collect them later. But at the beginning, let's focus on one single organ. Now, when you have this organ, you have to think if you would like to, what kind of conditions, under what kind of conditions this organ will be when you would like to study it. So do you actually want a proper functionality of this organ because you would like to use it for site toxicity screening, for example, and you actually want to establish a proper, very close to human physiology organ on a chip. Or actually, you would like to recreate a disease biology on this chip and use this chip to better understand the disease. Or actually, this is really far end, have this disease model, but actually use your proposed therapy or a repair mechanism on this disease model. So please think now about your conditions. So you have organ, you have conditions. Now, once you had your organ, actually you have to go a little bit smaller, right? Because chips are really small, so we cannot build a whole organ. We have to find something smaller. This something smaller is called basic functional unit. So every single organ has this basic functional unit with its own basic morphology and basic physiology that actually represents, let's say, a miniature version of your organ. This we will be trying to recreate on our chips. So, for example, for a muscle, it will be a muscle fiber. For that, it would be a villus. It can be an epidermal unit for skin. So please think about this basic unit within in your mind. I hope this is not very difficult at the beginning. Think about the morphology now, about really a 3D structure of this basic unit. What kind of cells are there? Which cells are close to which other types of cells? Which cells are kept in place, and which cells are actually flowing, for example, in your vessels? What are the proteins of your external matrix? This all will be relevant, and probably maybe at some point you will have to go back to it to match those conditions on a chip. When you're thinking about the chips, you want to be as close as possible to this morphology of the functional unit. Same goes with functionality. Functionality-wise, we want to be as close as the real human tissue. So we are still thinking about this basic functional unit, but now we're thinking about its functionality. And here, one of the basic functionality, but that it's very often forgotten, but very important when we are actually recreating organ on a chip, is this cellular communication. So which cells are under what type of communication with what other type of cells. So think about how the cells are talking in your functional unit, how the signals are flowing, and then think if their basic functionality is actually stretch or contraction, like muscles. The blood filtration for the liver, substance exchange, some kind of transmission. So think about these functions because this is exactly what we would like to have on the chip. Okay, so I think that was really easy. We call it context of use, the biology part of the chip. I think this is something easy to establish. You probably work with those things for years. So when you have this biology well established, and when you know what you would like to study, we can start to create to actually start researching our platform. So here I cannot give you right now an easy fixed solution. What type of platform should you use? I mean, of course, use Dynamic 42, but let's be real. There are many solutions there already on the market. There are many solutions there also in academia, in the basic research. So this is the step when you will have to research a bit. But the first thing that you're supposed to remember when you start thinking about the platform is that the platform should suit the biology, not the biology should suit the platform. I think if you work with cells, you know how very dramatic they are and how easily they die. You cannot force them to do or live on something that they're not meant to be in cooperation with. So that's why it's so important to think about the biology at the beginning and only then about the platform. So you can go ahead and start thinking about should this functional unit, is it actually okay to use a monolayer of the cells? Should this monolayer of adherent cells would represent the basic units? Or would you rather have, yes, monolayer of adherent cells, but few of them, right? So for example, using our channels, we can have a monolayer on top and monolayer on the bottom. what kind of cells should be under suspension? Should actually there be any organoids buried? Should there be any hydrogel? Because actually external matrix plays a very integral role in your basic functional unit. There are different also designs of channels. You will have to always think, okay, if this design that I'm seeing right now that is proposed by some company, is it actually fitting to my basic functional unit? And it's the same when we are thinking about the functionality of this organ. If the chip that is proposed to me actually can recreate the functionality that I would like to study. So here, you just have to look around, find your research, find your companies of interest. Go ahead and ask them what are their thoughts on the chip that you would like to have. And I hope you will. It will not be very hardworking for you. But there are many different solutions already out there, so I really suggest that you go for one of those. One thing when we are choosing the platform, we also have to think that this platform should fit, primarily should fit our biology, our basic functional unit. But secondly, they should fit the readouts that you run in your lab. So of course, when you have very complex tissue, you put a lot of money, effort, time into creating a very complex biology. You don't want to run into any issues when you finally have to analyze them, right? So please, while you're picking the platform, think if this platform is good enough or easily accessible for your readouts that you run in your lab. So here we are actually thinking about some golden standards that you would like to use with your organ, but also when you will be thinking about your new research, experimental design, and so on, what kind of readouts would you use? And if this is possible with your chips. So at Dynamic 42, we are actually very happy that our chips are very diverse. You can do a supernatant sampling, which is very easy with the microfluidic chip. This is always a no-brainer, I would say. We are doing the live cell imaging on a chip and also do the fixed cell imaging on a chip. We are running enzyme activity reactions also on the chip, for example, to study cytotoxicity of the liver. What is very, I think, unique thing for our chip, I really think it's personal. Like it was really one of the stuff that I really believed in this chip when I started to work on Dynamic 42. Is when I saw that you can actually recover your tissue from the chip. So I used to work with PDMS chips, and that was not possible. Once it was on a chip, it was always in a chip, and you have to work around it. But with Dynamic 42 chips, you can simply cut the top foil, which is just a foil, a transparent foil on the top with this scalpel. Cut the membrane that the cells are attached to, and just move them. And if there is any readouts that it's stopping, that it cannot be used with the microfluidic chips, you can simply cut out your cells and have them outside. So I think this is really a great application, very creative. I would say in this design of the chip. Okay. So we have our platform that is fitting for our functional units. We check that our platform is made for our readouts. I hope you are still there with me and still adding those steps in your mind that you are actually checking that yes, maybe some platform is actually possible to do a fluorescence imaging, for example, with your special 3D sample, and it's actually feels reasonable for you. Okay, so the next step is the cell sources. This is actually an easy step because I think biologists have it really covered with this knowledge of what kind of cells we want to use. So of course you can with our chips you can use stem cell derived cells. primary cells, cell lines. We are working on all of those type of cells. With on the chips, you can actually think about not only co-culture, but 3 or quad cultures, so really having many different culture of cells being together in during the cultivation. Yeah, I hope you can handle the cell sources quite well. And the last thing, when you are thinking about this technology, that it's fit for your tissue, your model, and bringing this to this next level of matching it better to the human physiology, is flow. So we have this flow conditions, which I think in an organ-on-a-chip world, they are divided into two groups. It's either pumps or this gravity flow, when you have your chips working in the on the rocker. I think this is the name, the rocker. And then you have the gravity-driven perfusion in your chip. We strongly suggest pumps, especially the peristaltic pumps, which are better mimicking the blood flow in your system. With the peristaltic pumps, the system that you are actually using while the chips are plugged is closed. So they're easily to removed and easy to switch the settings. So you can also try different flow conditions. And I mean, do try different flow conditions, because they have effects on your culture. And do you have flow conditions, because we can see from many different cells, the flow conditions have a great impact in their functionality or even morphology. So definitely having an easy to operate, easy to fit to your incubator system, a pump, a peristaltic pump, should greatly ease up your daily work with the chip. Okay, so we created this fit for purpose. We have a platform that we think that it will be fitting, suitable for our biology. We think that actually all the readouts that we would like to do with our model are possible with this with the chip, which shows the cell sources that should be suitable for our representation of the model. And we have even flow conditions that we can try out and start to play around while on organ-on-a-chip. Okay, so you have biology, you have platform. It starts to get serious, and it starts to get first test going. So we suggest not to go immediately into your biology, but we are recommending to go easy with it, and first start simple and then go complex. So first of all, really think about your setup, where you are starting. So for example, where you're currently in the lab. For example, your cells are currently on a plate, and then you would like to move them to the chip. There are still things that you should consider and be mindful that there are different if you want to move the cells from one environment to the other. And let's be honest, the cells will be dramatic, and then they might not like some material that they are on. So we are very happy. We tested our dynamic 42 chips with many different cell types, primary, non-primary, well established, just as isolated. Everything that we can get our hands on, and we never had a viability issue on our chips. But still, just to be on the safe side, please have a first test just to see if your cells are viable on our chips. So, and really just start simple. One cell, okay, they're alive. Then you add the second one and really start doing this step by step. So for the first test, yes, I would like you to think that you have to see if your cells are actually happy and alive on your chips. So to establish that actually your model is the base of the model, the most simple setup is actually possible to do, reproducible, and actually can give you a really great science. So think about the definition of your model. Think about the experiments that you would like to do, and try it while you have everything in your lab. Maybe it's also a good idea to try to think about it again. So again, start from monoculture and the simplest setup, one cell type, then add the other one. Check if any artificial material that is introduced with the chips is compatible with your cells. And checked if, for example, the perfusion is tolerable by your cells, and if you should optimize the protocol that you are using on the plate, if you have to optimize it somehow for the chips. So, for example, here we have this very typical problem with the chip, that there's always what is always new is this perfusion, and you have to put your cells that were usually grown on a plate under perfusion setting. So first of all, I think if you never had this experience, I think it's a great idea to actually go ahead and choose a model that is established by a given company that provides organ on a chip, because usually then a perfusion rate will be provided with your chips, or at least a hint what kind of range you should test with your cells, not to start from the zero. And then you just have to just run the first test, just get started with those chips, seed the first cells, and then of course compare side by side the static conditions, the slow perfusion, and then also the higher perfusion. So here on the photos we have two examples. So on the top you can see the example that did not work, and perfusion that was used on those cells was too high and the cells were washed away. And at the bottom, you can see the cells that the perfusion, actually, this is a suspension of endothelial cells in the gel. When kept static, these endothelial cells did not create any vessels, but once perfused, they actually created a very, very nice yeah, network of the vessels. So yes, so again, also during the perfusion, while you're using the perfusion, establish first one cell type and then add the other one. And if you have any more questions about, okay, how to set it up, something is not working, you can always use the literature as an orientation. But if you work with us, you can just simply call our sales or the scientist who is helping you to establish those setups. So I think we will be very happy to help you. We of course want to run this technology successfully. Yes. So when you're thinking also about first test with the chips, sometimes it's not only that you will be putting your cells from a plate on a chip, but sometimes you have some extra conditions that comes around it. So for example, here there's an example of the top picture shows an example of the spheroids, or we can see cells in hydrogel on the chip, or we can just keep them under perfusion when they're creating FPD structures. So sometimes you also have to check this condition. If the cells that were cultivated always on the plate, they are actually able to do a very nice spheroids before you put those spheroids on a chip. So really also think about the steps in between that you may have before you actually put some stuff on the chip. Sometimes it is just easier to test something very quickly outside of the chip, just to check that it's actually compatible, and then run it on a chip to create this complex biology. So, for example, if you're worried that your cells in a 3D culture on a chip, there might be a little bit too much, it might be a little bit complicated, and you may get lost, first run your 3D culture outside of the chips, and then integrate it on the chip. Okay, so you started to run this experiment in the lab. You had this first experience with the chips. It seems that it's going well. No air bubbles, so congratulations. But of course, when you start thinking about those experiments, there is a very important, very boring, but we have to do it step, which is to validate. So we have to trust what we see. We have to trust that those conditions are actually supporting the functionality of your cells, that they're actually providing those benefits in the cells that they're growing inside. They're actually happy. So yeah, we have to run replications. We have to try, for example, different flow rates to see that yes, when we are flowing under a higher condition, under higher flow, actually this stimulates even more the reconstruction of the 3D culture. So yeah, just ask yourself, is it alive? Does it flow? Can we see the signal? Does it repeat? And just validate your system. So with this, we have, I would suggest cell viability and attachments assay, something, those kind of readouts that I like to call a basic readouts. They will be giving you an information if your cells are, okay, not very scientifically, but happy. And they are not yet there to provide you a scientific modality. So bear with me, it will come, but at the beginning, please focus on this basic establishment of happy cell. So we have the cell viability, the medium flow, and the bubble control. That's actually this flowing in the system that you establish. Does it introduce some air bubbles in between, or actually like our dynamic 42 systems, it actually has a bubble trap in it, and the bubbles are really nicely, tightly under control. We have also our baseline readouts. This is what I said. This will give you the answer on how happy your cells are. And of course, please check and make a reproducibility check that with different runs of this of your models, the cells are repeatedly happy, and then you will know that your model is actually good and ready to move on. Always compare to conventional controls or literature. Or call us if you have any questions. Yes, we have the next step, which is kind of like finally, and kind of it's finally starting to be more excited and more interesting, which is getting more complex. So from these first steps, let's say baby steps, starting small, starting simple, we're actually adding more and more complex, and we are really getting into this human physiology. So this is the step when you actually start adding your immune cells in perfusion. You established your endothelial monolayer, one monolayer of the cells. This is the time to add the second type of cells in the channel, for example, as immune cells in perfusion. Please check your experimental conditions and check if the cells react. So we are no longer operating on those basic readouts, but yeah, what we are trying to establish these readouts that we would like to run to make our science and the novelty and publications, all of those stuff. So please make the first test with, for example, media with your drug that you would like to test. See if the cells react. Please introduce not if you are thinking about the disease model. Please start with healthy organ, establishing a healthy organ at the beginning, and then reintroduce the diseased cells or induce some pathology conditions. So for example, first create this basic model, but then put this model in low oxygen rate or higher perfusion, the pathologic perfusion that you would like to test. Okay, so from the two monolayers, you can also switch to 3D culture. So I think you have here two examples from our Dynamic 42 applications. So from very basic design, two monolayers in two channels, we can go one monolayer, but in the bottom channel, we actually have spheroids enclosed in microcavities. And then we can induce also immune cells in the top channel and see how these immune cells are migrating to the bottom channel to test, for example, the CAR T cells for your cancer model. The second application that we run is intestine model. So in the top channel, you have a monolayer of the cells, again, perfused with the immune cells. On the bottom, you have under perfusion a CACO2 cells, which creates a 3D microvilla, which is the response to this perfusion. And then we have a special TR sensor chip that we can produce for you. That is established also for the intestine chip to actually see how tight the endothelial barrier is. And if, for example, under pathologic conditions, this endothelial barrier is less stable. Okay, so this is really like, we're talking complex. So please remember that when we're talking about complex, similarly to the first baby steps, we have to validate. So of course, that's why it's important to start simple, validate those simple steps, and then go complex. So at the end, when you know that something, while you're in a complex step, while you are there, and then you are facing some problems, this is connected to this get more complex steps, not the first steps at the beginning. Those are set, and you know that they're good. Then you can go more complex. Okay, so we have the last validation. It's kind of like, this is the end, guys. Like, if you finish this step, you have a well-established model that you can run your science on and be an amazing scientist, okay? So here, for example, you can see we used our liver on a chip to test the site toxicity of a drug called trovafloxacin. And here we can see how sensitive our chip is, because we could detect a cytotoxicity at the very low concentration of 10 micromolar of the drug under perfusion, and even higher cytotoxicity under 20 micromolar. Now, trovafloxacin was actually successfully cleared through the clinical trials without any signs of liver toxicity. And it was introduced on the market, and only on the market it was reported to damage human cells, human livers. So we really want to avoid that kind of situations with the drugs. And here it's a great representation that with our models, if you would run the trovafloxacin tests on our models, you would see that kind of damage. Okay, so we are in this validation steps. We are using primary tissue for comparison, how closely we are to this endpoint human physiology. We are checking, we are looking for the confirmation of accurate model function. Yes, it actually recreates all the important steps that we would like to have in our basic functional units that we were thinking about at the beginning. That actually we can reintroduce the drugs, and then these drugs have an effect on our tissue. We have a key events for our pathology of the disease model, and we can say how sensitive or specific our model is. How sensitive, how little of the drug it needs to actually see the difference, and how specific it is, how much of the liver on the chip actually is. Okay. And then at the end, it's like, really, you are the master of this organ, and I think you're ready to go to work the science for it. But you know the reality of the research. So very often it is not very linear. This last step, we call it proof of concept, but it's rather going back and forth. So it's a process. So I hope you will stick in there. I hope you will go to this last steps and you will make your desirable research. So all the best of luck for you guys in here. Since we'll be sharing this presentation at the end with you, I also kind of compile a summary slide at the end where all the steps are taken together. So I hope you followed me through this for this presentation. You added some steps, and now as we are going closer to the end of the presentation, we can start just slowing. So you can think about the steps that maybe some problems to you, and I would be very happy to help you to go through this steps. So very quickly, just summarizing, you start from context of use, you define your biology. Then when you have this biology in your mind, you look for the platform and all the things around it that actually fits this biology that you have you have in your mind, not the other way around. Biology goes first. And then once you have biology, you acquire the platform and you're ready to go to the lab, please start in the lab small, start slowly, start simple, and step by step get more complex. This will make your success more probable. But you know, if you are feeling that you actually need a little bit more hands-on experience, you can join us in Vienna. We are running academy, which is 2 days. You will be with our scientists in the lab, and you will be really seeding the cells. We will be learning about the chips. You will have the opportunity to really touch them, pipette on them, cut them open. And then the second day, it's all about the readouts. We'll learn how to use it in the lab. We have also academy option online for those of you who do not live in Germany. We also have we recorded our scientists, and we run a platform to accommodate to share the knowledge about the chips also online. So I think with this, I can finish my presentation for today, and I can take all of your questions about this step-by-step guide, and I hope it's something that it can be helpful for you to establish the organochip research.
Thank you so much, Marka. Well, now we start with questions. But before I move into it, I would ask Bianca to turn off her slide deck so she can actually turn off her sound as well, which I have to apologize. This was the issue of the sound problems in the beginning, but this should be solved now. So just before we move into into the questions, I typically start our last call because we would really like to know how you like this webinar. Because if this was good, we'd like to do more of it. If it was not good, we can definitely do better. Okay, so now questions. We already have a first one here, which is from Lucia, which would be, do you have a general model for cancer or also organ-specific cancer chip?
Yes, it's a great question. I think it's one of our newest chip. We do have cancer on chip. It's our actually spheroid on a chip. It's our 3-channel spheroid where we can sit the cancer cells as a spheroid under the flow. We used this chip to establish pancreatic adenocarcinoma. Thank you. And we are still running it for our glioblastoma studies. So we do not cover all the cancer types, but we will be very happy if you can establish another cancer type on our chip, and we can support you with all our knowledge around the cancer type. I think I can also share a little, it's more like a gossip, that we will be providing in the future a product that supports actually recreating a true tumor environment on our chip. So if you are really interested in studying how the environment of the tumor is affecting the tumor itself, then you can stay tuned on our LinkedIn, and we can also make sure you will get this information once they're available.
That was definitely very much hot off the press. Okay, so we've got another question, which would be, where can I look if I want to find out which platforms are developed, or let's say,
Really good question, because I think it's quite problematic, because there are so many different platforms that it might feel really overwhelming when you start. I guess just we're all starting just by Googling. Okay, I want to do a liver on chip. Let's Google this out and let's see what's possible in there. And I think you can be overwhelming by those responses. But I think the first step is really to just have a very specific definition of your biology. So again, you look for the diseased liver, or actually a proper representation of the liver functionality. This should already guide you into what applications might be suitable for you, and what not. And of course, you can just see what is there, start digging in the scientific publications, in the websites. But I think it's also really good to just go ahead and establish a meeting with the sales from this organ-on-a-chip companies, because within 1 hour you will have all the information that you need about the specific chip from their companies. And I think this is the easiest way to really understand if this is the chip for you or not. So just go ahead and try to call those guys. I think they will be very happy to talk with you.
Thanks, Joanka. We've got another one from Edwin, which is, can you elaborate on the role of fluidics and sensors in monitoring the functionality of an organ model?
So the sensors in organ-on-a-chip, I think they are very helpful. Because they really can give you a real-time data on what's going on in your chip. And sometimes when you are really digging into some specific details, these details can be very dynamic in this live model. So I think, yes, at some point in this more complex, more established models, I think getting familiar with the sensors can help you. But again, bear in mind that you remember what kind of question you would you like to answer, because there are also different types of sensors. So would you want to, for example, if you are studying the oxygen levels, if you would like to have the oxygen in the media, or actually you are really interested about the oxygenation of your cells. So similarly to the platform, please research the sensors while once you're thinking that it might be very, very helpful for you. And then sensors are usually integrated in the chip. So it's not an additional to already existed chip. You will have to buy a chip already with the sensor.
Cool. So we've got actually 2 more questions by Xing Xiang. The first one is, are there any working examples of gut-on-chip and anaerobic culture of bacteria and fungi?
Yes, they actually have an established model of our intestine on a chip. It's one of my favorite models, because we can clearly see that when you will keep the intestine chips in the static conditions, they are remaining really, really flat. But once you put them under flow, they start to recreate the microvilli. So actually, it's a very important model, I think, to have actually on a chip, not on a plate, because we can see a really great impact of this perfusion on the cell's morphology and functionality. You can go ahead and find information about our application on our website. We use this chip to study the fungi infection in our gut. It was so advanced that we could also analyze the behavior of more virulent and the candida that was resilient to antibiotics, more resilient to the antibiotics treatment, and how they looked and how different they were in the morphology, and we could match the morphology to more virulent features of fungi. So it was a very successful study. Yes, and but for the anaerobic conditions, we could definitely virtual. And here, I think there will be a no-brainer to actually use our oxygen sensors to actually really trace in the real time when you are reaching those anaerobic conditions inside of the chip of your media. I think with our chips, there will be no problem because we are not using also the PDMS. PDMS is oxygen permeable, so I would not recommend to go with those chips when you're thinking about no oxygen conditions. But I know our chips are not that easily air permeable. But for more detailed questions, you can go ahead, send us an email, and we will make sure, for example, that you can, with our gut team and people who actually establish those cells and work with those models on a daily basis to really talk scientist to scientist and how it goes.
Sounds good. And I've also added a link to the chat, which might be quite relevant to you. So this is one of our customers from the University Hospital here in Jena, and she's working on intestine and chip with Clostridium, I think, in hypoxic conditions. So yeah, watch that. I think it might be quite relevant, and also look at the publications of that group Inspire at the University Hospital. I think that might be very relevant to you. The second part of the question was how to balance the gas conditions for cell culture and the anaerobic conditions required for bacteria. And I think in part, Bianca, you probably already answered it. Is there anything else that you would like to add?
I maybe to those there was some question about the gas conditions. What was the part?
So the question was how to balance the gas conditions for cell culture under anaerobic conditions for the bacteria.
So I think that the easiest one is to run some part of your culture in low oxygen incubator, and I think just keep those cells in there. And I think if your system fits into this incubator and also allows to have a media partially exposed to this environment without oxygen, that would really benefit those systems. But I think for more details, I would go ahead to some scientists who actually work with our gut on a chip. So if you have like more specific questions about it, and I think you are really like into getting complex, I'm almost there. I think you can go ahead and let us know that you are interested in those anaerobic conditions, and I will make sure that you will get in contact with our microbiology scientists.
I apologize, my sound wasn't on. I said we've got time for more questions. And the one I'd say is like, how do you determine the optimal flow conditions for the different organ models? Great question.
Really good question. I think the only answer is you just go ahead in there and try. But the good side of it is we also in Dynamic 42 we went there, we tried, so you don't have to. So if you go ahead and use our application notes and the established systems, it also comes with the recommended flow conditions. If you actually want to establish something very new and never tested, we would still we would like to support you, and we will give you the rates that we would say have a feeling that it would be arranged to start working with it. But at the end, if you do not have this pre-tested conditions, it's also a question. Maybe there is some information also in the literature, right? So this is also I think a great source to go ahead and check. But if you do not have anything to work on at the beginning, you just have to go on and try. And you will see if there is too high flow, it will be just start washing out your cells. So you can clearly see it under the bright field. If the flow is too low, there might be no reaction at all. So you just have to go ahead and start trying. And I think this is the moment of this first steps and validation moment when you are starting to get to know to your system and trying to understand what kind of conditions are good and supported by the cells and or not. So I would not be afraid. I would not cut completely this step, and I would just try to understand your cells a little bit better. But I promise you, if the perfusion is too high, you will immediately see it. So I'm just this.
Excellent. Thank you so much, Joanka, for presenting today. I hope that was useful to a lot of you and that you liked the webinar. As I said, you will receive the recording after this, probably sometime around next week, once I get around to editing it. And yeah, so we hope you join you for one of our next webinars as well. And until then, I wish you a good rest of your week. Goodbye.
Thank you, guys. Bye.